Hi guys, today we are starting our unit on genetic engineering and biotechnology. Some of the labs that we're going to be doing really emphasize a few of these procedures more than others, but here's four examples of things that have been done through genetic engineering and biotechnology. So the top left picture that we see is of a chicken that was specifically bred to be hairless or featherless. You can also see that it also has some pretty meaty muscles going on in there, which would translate to a better chicken product. Like that wing right there looks pretty tasty. Um, in the top right, we have a mouse with an ear growing on its back. This was achieved through stem cell transplant, where the cells were originally not programmed to do anything in the body. They were just available and hadn't been determined what they would be yet, and were triggered to turn into an ear. And since the DNA in your body it contains all of the genes, and just certain ones are turned on in different locations of your body, the stem cell was triggered to turn on the genes that create the shape of the ear, and now the mouse has an ear on its back that could be potentially grafted onto its original DNA owner at some point. The bottom left and bottom right all are of the same idea of taking a gene from one animal and putting it into another animal. Okay, so in this case, we're looking at fluorescent proteins. The one we're specifically going to be doing is from jellyfish, but inserting it into another animal where under UV light, this cat and these mice glow green. So what is biotechnology? Biotechnology is using a biological system to make products. It's technology based on biology, agriculture, food science, and medicine. Usually we refer to genetic engineering and cell and tissue culture technologies when we say biotechnology. However, this could be encompassing a lot more procedures for modifying living organisms according to the purposes that we need them for, going all the way back to domestication of animals, cultivating plants, and improvements to these plants and animals through breedings that employ artificial selection and hybridization. Basically, you're finding the ones that you want to breed together. Let's say two plants that have some amazing fruit so that you can get more amazing fruit in the offspring. So that's artificial selection. And hybridization is mixing two types of animals together. So hybridization. Spider goats. Well, not really a goat that can build a web, but scientists are working on isolating silk-producing genes, like from spiders, very strong silk, like Spider-Man, to insert them into goat embryos in the goat's genetic code so that the goat would produce silk in their milk. If you have a lot of silk, then you could make things like bulletproof vests that are very thin and light, artificial tendons, and computer chips. Instead of using silicone, using silk, fiber optic surgical tools. All of these things are possible through silk fibers and goat's milk. So that's an example of hybridization, taking a gene from one animal or species and putting it into another species. Scorpion cabbage, another one. Toxin isolated from scorpion venom that's inserted into the DNA of a cabbage that will be toxic to bugs and the pests that want to eat the cabbage, but not to humans. So it's kind of a natural form of pesticide. You're not spraying a chemical on the cabbage. You're actually giving it a gene to be gross tasting to insects and kill them. So what is biotechnology? We're using the tools of biology to create new products or to change living things. You see a guy holding salmon there. Salmon's what I did my research on, so I'm way familiar with those guys. These Atlantic salmon are about the same age, hatched at about the same time. The big one was genetically engineered to grow at twice the rate of a normal salmon. So that scientist on the left holding a micropipette and a centrifuge tube, it looks like, used some kind of methods to insert genes or to trigger growth hormones inside the larger salmon to make it bigger and tastier. Scientists aren't trying to be responsible with these, though, or irresponsible with these. They're not like letting these giant salmon out into the wild to fend for themselves. Usually, genetically modified fish have to stay in fish farms where people will have them in tanks, breed them until they're big enough to harvest, and then make you some fillets. Okay, but we're using biology to create new products or to change living things. We have been doing this for generations. Artificial breeding is creating new breeds of animals and new crops and plants to improve our food and lives. You see in the black dog on the left, that's a Labradoodle. That is a dog that was specifically created to have the qualities of a poodle and the loyalty of a Labrador, but also be hypoallergenic. The picture on the left, kale, cabbage, cauliflower, broccoli, Brussels sprouts, all of those were artificially bred from the exact same plant to be all of these different plants. So not normally thought of as biotechnology, agriculture will fit this definition of using a biological system to make products because using what we know about plants and how they were domesticated and cultivated for a food source, it's probably one of the easiest examples of how we have manipulated plants. Okay, so agriculture has theorized to become the dominant way of producing food. And the processes and methods of agriculture have been refined by our mechanical and biological sciences since the dawn of our cultivation of them. Through early biotechnology, farmers could select the best suited and highest yield crops, or the most producing crops, to make enough food to support a growing population. So, for example, a corn plant that has 5 ears of corn per stalk versus 2, you're going to want to breed the 5-ear stalk so you can feed more people over time with less land and less water. Specific organisms and organism byproducts were used to fertilize, restore nitrogen, and control pests. So, throughout the use of agriculture, farmers have inadvertently altered the genetics of their crops through introducing them to new environments and breeding them with other plants of the first forms of biotechnology. Pretty cool. So one other thing that we know is we can trace the genetic similarities between all dogs, or Canis lupus familiaris, back to the gray wolf. So people first domesticated the gray wolf several tens of thousands of years ago, and they provided early humans with guard animals, a source of food, fur, a beast of burden, like think dog sleds. And this process continues today. So intentional crossbreeding of dogs, creating new designer dogs, okay? Yes, at some point, the poodle was bred for a specific purpose, and the chihuahua was bred for a specific purpose, but not the same purpose. Keep that in mind. Carl Vila, who has conducted the most extensive study to date, has also shown DNA evidence that has ruled out any ancestor except the wolf to our modern day dogs. So people thought, well, a wolf, but maybe a coyote could be a dog ancestor too, because they're similar. It's smaller, so maybe that could explain some of the smaller breeds. But Carlos Vila did genetic testing and realized that the only evidence was for wolves being the ancestor. Remember that magical plant I talked about? Descendants of the wild mustard plant, the yellow flowering plant on the left there, were bred in different ways to make all of these products: kohlrabi, kale, broccoli, Brussels sprouts, cabbage, and cauliflower. Okay. Now, if you think about broccoli and cauliflower, they have similar shapes on the top. Broccoli's green and cauliflower's white, right? Broccoli grows on a stalk and cauliflower grows more in like a ball, a brain shape. Brussels sprouts are a lot like a cabbage, but just tiny, right? A lot of different leaves folded over one another. But if you think about it, the head of a broccoli has like tiny little baby Brussels sprouts all on the top, right? The little leaves of the broccoli are kind of like the Brussels sprouts, just really tiny. And through breeding these plants differently, by breeding so that, let's say, the leaves on the sides become larger for the Brussels sprouts. You can see the modified trait is listed below. The stem for the kohlrabi to make a big bulb at the bottom. The leaves for the kale. Notice in the original mustard plant there are leaves, but they're not veiny. They're not big and leafy like in kale. So they were selectively bred to have larger and larger leaves over time. The flower buds in the stem are selected for in broccoli, so they have that nice tree shape. Lateral leaf buds on the side of the Brussels sprouts, terminal leaf bud for the cabbage, so the cabbage is like a giant Brussels sprout at the tip of the plant, I guess. And then, of course, the flower buds for the cauliflower. Another plant that we have definitely genetically modified is corn. On the far right, we have today's corn. On the far left, we have the ancestral teosinite. which I guess you can kind of see a little bit of corn kernels in. But it was a group of large grasses of the genus Zea found in Mexico, Guatemala, and Nicaragua. And by selectively breeding that over time, you can see in the middle picture, you can breed it different ways to produce different fruit, essentially, which is the little kernels of corn. Make it tasty. Those are the seeds. And so the more you breed them for the seeds, the better looking your corn is. I know there's a lot of different genetic modifications of corn in this day and age, and I'm not going to get too much into GMO corn right now, but you can see that there's definitely been breeding from the ancestral teosinte to the modern corn. And that was done without genetic engineering, obviously, since we didn't have that back in the day. But it was done just through breeding bigger and bigger plants together with better and better seeds. So, can we transfer a gene from one creature to another? Yes. Okay, on the left side, we see the jellyfish protein. And this is the lab we're actually going to be doing, not into mice, but into bacteria. Jellyfish protein in transgenic mice. Transgenic means you're just taking the gene from one organism and putting it in the other. So we isolate the glowing green gene in the jellyfish, and we insert it into the mouse's genes, and voila, mouse that glows. Okay, and the parts that are glowing brighter are the parts that don't have hair covering it. If it was a bald mouse, we would find that it glowed evenly. Luciferase is a firefly protein, makes it light up. And you can see that pig looks like it ate a highlighter, but it's actually the protein from the firefly right in that pig that you can see. Ways that we use this for medicine. We allow organisms to produce new proteins. We've harnessed the power of bacteria to produce human insulin. So for people with diabetes whose pancreases can't produce their own insulin, Humulin, which is actually made by Genentech in 1977, figured out a way to make human insulin in bacteria and also human growth hormones. They were the very first, actually, to produce genetically modified medicines. So basically we have a giant vat filled with bacteria. They secrete the insulin out into their environment. You purify it, put it into a medicine bottle, and there you have your insulin and growth hormone. We also use genetic engineering for food. Not all genetic engineering is bad. Some of it could have other effects that we don't see right now. But there are some things that are currently approved for the U.S. market. So, for example, a transgenic rice plant has been developed that produces yellow grains, you can see in the bottom right, containing beta-carotene. Beta-carotene is used in your body to make vitamin A. Currently, about 70% of children under the age of 5 in Southeast Asia are deficient in vitamin A. That leads to vision impairment and increased disease rates. So by perfecting this golden rice and making it readily available, we could see an improvement in the nutritional value of rice in Southeast Asia. You see the middle one, extended growing season for fish berries, which sound awful, but strawberries that have a gene from flounder that help them resist cold. And then, of course, protecting crops from insects, BT corn. The corn produces a toxin that kills the borer caterpillar, but is not harmful to us, or it doesn't kill us at least. Okay, so this is examples of genetic modification. Kind of like the scorpion cabbage we talked about before, where the cabbage produces the scorpion toxin. We use genetic engineering for DNA fingerprinting as well, and that's another lab we're going to be doing. Crime scene investigators and paternity test people will use DNA fingerprinting to figure out who parents are, find criminals, rule out suspects, all sorts of stuff. Everyone's DNA is unique unless you're an identical twin, and we can use this DNA fingerprinting evidence to, within about a 3 billion chance, 1 in 3 billion, we can narrow it down to it being you. So there's 7 billion people on the planet. If we have a 1 in 3 billion certainty, unless that other person happened to be in the same place in the same time as you, cops can figure out that you were that person that perpetrated whatever crime they're looking into. So the tools that we're going to be using, for our first lab, we're using micropipettes mostly. And these are kind of like regular pipettes, except we measure small quantities of liquid. There's a dial on them. We'll go over how to use it, but usually under a milliliter of liquid is measured in micropipettes. And there's a great technique for using them. And they're really fun because you get to shoot the tips off, and it's a good time. And there's also gel boxes, which help us to separate DNA fragments that we create. And we'll do a lab with that later on. Micropipettes are used for measuring very small amounts of liquid, and they're used in almost all biotechnology research and laboratory work. So this is one of the skills that's going to be really great to have, and it's really good for you to build on throughout this unit. Well, I hope you got a good idea of what biotechnology is and what we can do. And I hope you're excited as I am for continuing this unit. See you tomorrow.